vaccine design, molecular modeling, molecular docking, and molecular dynamics simulation, integrated biology, genome and proteome analysis, tool development, and so on for preparing you for the designing project, conducting research and publishing paper, arranging the seminar, presentation, and competition to generate the future researcher. So now let's know about our honorable advisor. So here, this is Noore Sharaf Nawar Samia ma'am, the most energetic and passionate person in the Biopc. She has been working for Biopc from the very beginning and hardly working for the development of Biopc. And Samia ma'am has completed her bachelor's and master's from BRAC University, and currently she is working in ICDGRB as a research assistant in both the virology and genomics labs simultaneously. And she has vast knowledge and expertise in cancer research, vaccine design, and genomic epidemiological tools. And we are so glad to have such gems advisor and trainer with us. So here comes the most introducing the diligent and skilled advisor and trainer of the Biopc, other than Gagandeep Singh Khurana. And currently, Gagandeep sir is the PhD student at Indian Institute of Technology, Delhi, and as well as he working in the section of microbiology, Central Ayurveda Institute of Central Ayurveda Research Institute, Jhansi, Uttar Pradesh. And he has bioinformatics expertise and knowledge of computer-aided drug design techniques for the design and development of therapeutic molecules using docking, molecular dynamics simulation, and quantum chemical computations. The smartest and the most humble advisor of Biopc, Amit Kumar Datta sir. He has completed his master's from Japan. Currently, Amit Kumar Datta sir is serving as an associate professor in the Department of Microbiology, University of Rajshahi, Bangladesh. And he is very popular among his students for his kind heart and student-friendly nature. He has a strong research background in the field of bioinformatics. Till now, he has published about 35 papers in the reputed journal, which are very impactful in the fields of research. Many students are doing thesis and research projects under the supervision of Amit sir. Hope our Biopc participants will get a chance to work with Amit sir in near future. And we are proud to inform you that the most heavyweight and impactful profile, Dr. Faizul Azam, PhD, is the head of the project of the Biopc. Want to know more about him? And undoubtedly, this event was initiated by him, and he has designed all the research projects, and also he is managing Biopc financially and technically. Also, he will pay article processing charges for শোনা যাচ্ছে না।
Ma'am, I think your microphone is mute.
হ্যালো,
শোনা যাচ্ছে।
আচ্ছা, আপনি সবাই কি শুরু থেকেই শুনেছেন নাকি এখন অফ হয়েছে?
এখন অফ হয়েছে, সবাই শুরু থেকেই শুনছে।
আচ্ছা আচ্ছা, ওকে। ওকে। So, where were we? Dr. Fajr Azam sir is an assistant professor in the Department of Pharmaceutical Chemistry at Qasim University, Saudi Arabia. And he is very selective and sustenance in the case of his research works. Till now, he has published about 77 papers in the reputed journal, Hope you will have a good journey with Fazul Azam sir in future. So let's know about the Biopc event, Learn Bioinformatics with Biopc 2.0. This event has been designed considering the knowledge of undergraduate students. After completing the session, you will know about, everyone will get a certificate after completing the training session, and the top 10 participants will get a chance to work with the Biopc research team in several running projects. And Biopc research team will directly involve them for publishing the paper after completing the projects. And most passionate and enthusiastic participants will be joined as a volunteer in the Biopc organization. So here are the briefs of our running projects where the top 10 participants will get a chance to work. So in the next slide, here I am stopping now, and hopefully I have presented the session appropriately. So my time has gone away. Now it's time to transfer the mic to another. আমি অমিত কুমার দত্ত স্যারকে রিকোয়েস্ট করছি আপনার ভ্যালুয়েবল স্পিচ দেওয়ার জন্য বেসিক বায়োইনফরমেটিক্সে। আর আরেকটা কথা, ক্লাস শেষে কুইজ থাকবে, আর কোনো কোয়েশ্চেন থাকলে আপনারা কাইন্ডলি চ্যাট বক্সে দিবেন। স্যার যেটা ভালো মনে করবেন আপনারা কোয়েশ্চেনের অ্যান্সার দিবেন। স্যার, আপনি কি আপনার স্ক্রিনটা শেয়ার করবেন এবং আপনার মাইকটা আনমিউট করবেন প্লিজ?
Hello, can you hear me?
Yes, sir. Thank you, sir.
শেয়ারিং এর সমস্যা হচ্ছে বোধহয়। Yes, 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 yes.
স্যার
Now okay?
এর পাশে যে তিনটা ডট আছে এটার পাশে প্রেজেন্ট নাও এটাতে দিলে হবে।
এখন কি দেখা যাচ্ছে?
জি স্যার, দেখা যাচ্ছে স্যার, থ্যাংক ইউ।
So, dear participant, welcome to my session. Actually, this is very beginning part of this journey. Here, just I try to introduce you about bioinformatics, the basic of bioinformatics, and very preliminary things for bioinformatics. In order to gain the interest of you about the bioinformatics research and bioinformatics journey. Here, welcome to my presentation. Very, I think, very basic and preliminary things for bioinformatics. And as a researcher, I am also very beginner in bioinformatics study, because basically I work in molecular biology, and during my molecular biology research, I have to use bioinformatics, and now I try to do something by using bioinformatics, and at same time I also try to use the wet lab and আচ্ছা আমার কথাটা কোথা থেকে যেন রিপিট হচ্ছে।
জি, ডাবল হচ্ছে কথা আর কি।
স্যার আপনি মনে হয় ডাবল অ্যাকাউন্ট, দুইটা অ্যাকাউন্ট থেকে করছেন, ডিভাইস থেকে করছেন, একটা ডিভাইস থেকে।
আমার একটা অ্যাকাউন্টে, অন্য কারো কি লাউডে দেয়া আছে?
স্যার ছিল একটা, তাসফিয়া নামের কোন একটা অ্যাকাউন্টের ছিল, এখন সে অফ করে দিছে।
আচ্ছা, এখন বোধহয় শোনা যাচ্ছে। শোনা যাচ্ছে?
Yes, sir.
Okay, thank you. So, actually, I try to work with wet lab and the dry lab combinedly. And here I just try to introduce you bioinformatics. So first thing, you have to know what is bioinformatics. So bioinformatics is not so difficult thing. Is very simple. You have to know about the genetics and biological data first. Then this data, how to use and how you can find out the data. This is the process of bioinformatics. That means biological information management system or processing system. So firstly, your target is biological things which you use for your research, and these data are organized somewhere. You have to use this data from. You have to collect this data from there, and then you can manage this. You can analyze this, and this analyze you can present as a research result. And this is the fourth thing for bioinformatics. So here, if you concentrate here, we can find out some different uh, uh, disciplinary work. First, biology, because main source of the data is biological material. So we have to know about the biology. Biology means basically genetics or molecular biology, because here most of the data from come from our genetical information like RNA, DNA, protein, or other compound, and then we have to use this data, or we have to deposit the data or manage this data by computer science or computer computation study. And then this information technology is one of the thing we know that the information or like now communication system give our opportunity to use data from one same source or from different place. So this is the combination thing, or this is the combined field of research or science, where we can use the same data from different place, different source, and this data give our the opportunity to find out some new thing, and that we can apply for our wet lab, that can be give us actual thing. So firstly, we have to know actually bioinformatics many basically prediction. So firstly, we have to predict, or we have to draw the future line. So what can be happen, or how can we do it? So if we can set our target, so then we can go wet lab. It will be very easy for us. So we can save our time, save our money to success our research, or we can do something good for the nation or good for the art. So if you know, or if you just even look about the COVID-19 uh, research, so you can think the at a time whole world uh, try to fight or try to save themselves from COVID. And that time, every place of the world generate many data from COVID related data. They can analyze, they can research, or they can just generate data. So this data can be used for solve this problem, and very quickly many organization or many uh, research team can develop the protection system. So this is the one of the most benefit from bioinformatics. Then it can be save time, it can be save money from our actual findings. So I already say about these type of things. So actually, which is the main bioinformatics things. So here we can show this picture. The actual bioinformatics is the combination of biology, medicine, math or physics, and computer science. And this is also a multidisciplinary subject. So if we want to make our mind as a package for bioinformatics, we have to know all things. but it is really difficult. So we try to do this by using other people developed software. So this is the main component. So technology database, actually this database from our biological resource, where come from molecular biology or other genetical studies, and then analysis tool. So these tools are made by a specialized person who make the software, and this many company now make different type of tools for use as a bioinformatics tool and algorithm. Actually, this is accurately, or this is the absolutely come from the mathematical or computational knowledge and our computer power, computing power. So we have to know at least ABCD of computer for use the bioinformatics tool. Then we can start our journey. So now I just try to say the necessity of bioinformatics. So if we want to use bioinformatics, so we have to know why we use this. So the need of bioinformatics has arisen from the recent explosion of publicly available genomic information. Actually, if the communication system is not developed like this time, so this bioinformatics is not actually a valuable thing. But now we can find out a huge amount of data around us. So if we browse from our internet, we can find out many servers. They provide our huge amount of data, so that can be usable for research. So this is the main main advantage thing for bioinformatics. So now a huge amount of data available, like human genome project, like different database from deposited from different researchers, different genomic database, different protein database, and that can be usable for the research and gain a better understanding of gene analysis, taxonomy, and evolution. Also important for bioinformatics, and to work efficiently on the rational drug design, and reduce the time taken the development of drug manually. Actually, one of the big field of bioinformatics is drug design, especially if you try to design a drug, or finally you can make a drug, you have to go a very long way. So for reduce this time and save the money, this is the one of the process. So first use bioinformatics, then you can go some potential candidate by actual research, and that can be find out very short time result. And aim of bioinformatics actually the improve the content and utility of database, the main aim of bioinformatics, and develop the better tool of data generation, capture, and annotation. Also one of the aim of bioinformatics, and develop and improve tools and database for comprehensive functional studies, and develop and improve the tools of representing the analyzing sequence, similarity, and variation. Actually, this journey starts from this side. So if we want to analyze some similarity or variation, so firstly we need some deposit. Then we can find out some similarities or variation. This is the basic point, or very base point of bioinformatics. Is start from the similarity or variation studies, and the creation mechanism to support the effective approaches for producing robust the exportable software then can be widely shared. And one of the basic thing bioinformatics, and because now very good amount of software are available, and we can freely use this. This can be promote the bioinformatics research, and the bioinformatics research is very impactful for this reason. And field of bioinformatics, of course, molecular medicine, because
Sir.
hello.
Sir.
হ্যাঁ, শোনা যাচ্ছে?
Yes, sir. Sir, I want to say that if we can go the last slide, please.
নিশ্চয়।
Sir, third and fourth point and fifth point, can you explain again?
Okay. Third and fifth.
Yes, sir.
Okay. So develop and improve the tools and database for comprehensive functional studies. Actually, if you want to study the genomic data or molecular biology or gene function, so we have to know about, we have to gain knowledge about our gene or our protein. So for this study, firstly we predict something, or we try to compare our previous studies data. So you know, if we want to write something, we have to know the previous knowledge. So previous what thing will be done in this regard. So for this experiment, so firstly we have to know the previous data, then we analyze the data, and then we can predict this is the possible 2 or 3 thing can be happen. Then we go by that way. And this is one of the thing for bioinformatics, because previous data means gene, that means nucleotide sequence or RNA sequence or protein sequence can be say which type of protein and which their function can be done. So if we found this type information, we can go very easily in the for conclude our research. So this is the one of the most important thing. And for find out good result, we have to improve our tool. The tools mean different type of software. So every group of researchers done different type of release. Firstly, now we just are user of this tool. So and some of our expert member, that means especially who work very long time, they try to improve their tools. So and of course you have to know the basic knowledge of computing for improve the tools, because you have to develop, if you want to develop some software for analyze some data, so you have to know the how to develop. So this is mainly the work of computational computer background people, or statistical background people, who try to improve the tools which will be used, or improve the database. And another second, that develop and improve the tools for representing and analyzing the sequence similarity. So sequence similarity mean you have one sequence, you don't know what is this, or this come from which organism. So like you find out one microbes, and you collect their DNA. So or you found one DNA sample from one place where something happened, like a murder happened or something. So you try to find out these people, like for this is a example of a forensic medicine, or like new microbes identification. So you have to search this one. So how you can search? You have to go the depository thing or database, then you try to searching this one, and you have to for searching this one you need some tool. And here they actually say about this tool. So here in bioinformatics you have two part. Actually some people work and improve the tools, and some people are only user of these tools. So primarily we are user of this tool, but if we want to improve the tools, we have to go a long way. And the last one, the create the mechanism to support the effective app process. So for producing robust and exportable software. So this is another one. So so if you want to improve the tools, or you can make some server or software, so you have to change this one from other, exchange this one, or you have to share with others. Here, of course, here have some organization who commercially provide the software and take money. But actually, if you want to do something for the science, you have if you go, give it the free, it can be usable for all. This is another way. So some international organization and some national organization also give fund for this purpose, for widely used softwares, and we can also do this type of thing, because if we make one library from our uh, plants like Sundarbans or some other region, so this is not available for other, but we can make one depository, or we can make one server, we can collect compound from this plant, and we can make server, and we can open for it, so other people can use it. So this is also another way. So next one, the field of bioinformatics. Of course, the main field of bioinformatics is molecular medicine, because most of our research is come from molecular data or genetic data, and another is genetic gene therapy, drug development. Drug development is one of the most emphasized field of bioinformatics, and microbial genome application. So also it is used for different types of microbes identification, or microbes modification, or other things also, and crop improvement. So crop improvement, actually this is come from the plant molecular biology or plant genetic engineering. So it can be used for improve the plant, for crop improvement, actually for different type of stress, or by using this data for yield improving. And another is forensic medicine, for forensic analysis for microbes, or forensic analysis for, of course, forensic analysis for human also, and biotechnology, evolutionary studies, and bioweapon creation. These are the another dangerous thing for bioinformatics. So now we know science can be used for good or bad thing both. So this is also another thing.
Assalamu alaikum, sir. Sir, what is evolutionary studies?
Evolutionary studies is actually, we know our previous people make the systematic biology, make the comparison from organism to organism by their morphology. But we can now use we can compare their the organism by their genetic data, like DNA, protein. So if you compare their genomic sequence by bioinformatics study, it's the evolutionary studies. So what is the relationship from human and other organism, or what is the relation from different type of plant group, or this is the combinedly, or what is the relationship of Arabidopsis, like Arabidopsis recu group, or mice recu group. So it can be different stages, like gene to gene, protein to protein, from different organization. We can make the study for not only the whole organism, but also the specific protein or specific gene. And this is also very useful material for our all researcher evolutionary studies, because if you can write some paper in any organism or any people, most of the researchers add this part for their publication and for their research also. And where the bioinformatics help, so we already say some similar things. Actually, the experimental molecular biology can help it, because molecular biology basic data, if you know about the sequence, or you need the sequence information or protein information, it can be used, it can be helped by bioinformatics. in genetics and genomics. So genomics and genetics, of course, you need the genetic information, or you can find out some sequence plant or sequence animal or whole genome. This data you can be used for your genetic research, and generating the biological data also used for bioinformatics, because these have the system to deposit the data, or can be used the data, or you can collect the data and make another depository for your use. And another is analysis of gene and protein expression. Now many people work in different type of organism, and one of this RNA-seq or microarray. So RNA-seq is use RNA for gene expression, and microarray also another type of expression data, and this data will be deposited. And if someone published the paper, you have to give this data for deposit. And any researcher can use this data for their own research, because you can think the human have many genes. So my target is one gene. I can use like X1 gene, and for target of X1 gene, I can make some experiment for expression, and then I deposit the data. Another one use Y1 gene, they deposit the data. But now I target like A gene. So when I make the genome-wise expression, so whole genome expression data was collected there. So if I targeted one single gene, another single gene, so I can use this data and I can find out my interest gene from this research, and can make some prediction where this gene is expressed. This is also another, the analysis of gene and protein expression data. And comparison of genomic data, of course, the bioinformatics can help, because bioinformatics mainly one of the big thing for deposit data. So if I want to comparison the genomic data, so bioinformatics tool can be used for this. And last, the simulation and modeling of DNA, RNA, and protein. And this is also most usable thing for bioinformatics. So now make simulation. That means we create some artificial cell in our computer by this simulations program, and we try to find out by time what happened, what is the happen in DNA, RNA, or protein structure, and how can be protein and other compound are interact or make reaction with each other by time. So like we want to see it for 5 nanosecond, 10 nanosecond, or 100 nanosecond. So what can be happen in this time? So this is the process of simulation. That means we can make the artificial cell in computer. Then we can give the environment for our targeted cell, and then we can try to find out what happened by this gene. These are the simulation, and we can make the model of the DNA, RNA by our available model. An application of bioinformatics. So this is also actually same thing. So if I can find out the area, if I can find if I know our aim, and if I know our research field, so we can applicate. We can make application, or we can apply this thing in same area. So like molecular medicine, personalized medicine, preventive medicine, gene therapy, drug development, microbial genome application, waste cleanup. And if I say the major research area, so major research area is computational evolutionary biology. This is one of the most thing. So, and sequence analysis. So if we want to make the analysis and make sequence analysis for any sequence, so we need to go the database for compare or for find out our target. And of course, genome annotation for genome based experiment, it can be needed the database for our research. And identify the element of the genome, a process called the gene finding, and attach the biological information to this element. So if we find our genomic data, so we want to make the we have to find out this one and comparison the other data, then we can find out our actual name of our species or of our sequence. Then we can find out their biological information, and we can make the conclusion by this one. And another one, analysis of regulation. Regulation means if what can be done by this sequence. So if we have any sequence, so we know first DNA make RNA, then RNA make final product protein. So if and protein the work with a like a net. So it not a single thing. So it connected from each other. So like a orchestra. So it if one is increased, another can be decreased, or if one can be can stop, another can be what. So like a combination of each other. So this analysis also happened by bioinformatics. And in cancer analysis of mutation, also one of the major research area of bioinformatics, because if we try to find out an mutation, so we have to analyze the sequence. And another one is cooperative genomics. So if we want to compare two or more genomic data, so we have to need bioinformatics database. So another one is structural bioinformatics. So if you want to do some in drug design, so you have to know the structural bioinformatics. Structural means you have to predict some structure of DNA, RNA, or protein. So prediction of structure from sequence. So actually it starts from sequence. So if you have some genomic sequence, so you can make some structure. So it have some you here have very available of different type of server or software that can be that can be used for predict the secondary structure, or you can make homology modeling. Homology mean just based on similarity of this type of sequence, you can sequence because a new model of protein. And you also make the 3D prediction of any type of sequence by using the structural bioinformatics and analysis of the structure. So firstly, you can make some structure by your sequence, then you can analyze the structure, so you can predict the function of this protein. And structural, there have many database for structural analysis. So, and this can be happen by in drug design studies. So prediction of molecular interaction, docking, molecular dynamics, all can be happen by structural bioinformatics. And here we provide you some list of database. So first in bioinformatics, first you need the database. So a database means the depository of or information. So one of the here main we can provide you 5. So if we say more important 3 or 5 database, one of them is DNA Data Bank of Japan. This National Institute of Genetics. Another is European Bioinformatics Institute. Another is Gene Bank, the National Center of Biotechnology Information. Another and Uniport also another database, Swissport. And mostly important and versatile database is National Center for Biotechnology Information, NCBI. So we can say the NCBI is the biggest source of data for bioinformatics, and for not only bioinformatics or biological, any type of biological study, you can go there first, then you can find out the another location. And here have some special database like gene oncology for structuring, KEGG for biochemical pathway, PDB structure of protein, interaction database, and DBSNP, the database of genomic variation, and expression source or microarray data also available here. And among this database, the three major nucleotide data bank host of primary sequence data. So I said that these three data is the mainly the primary sequence data. So European Nucleotide Archive, ENA at EBI. This is actually EMBL. And another one is Gene Bank at NCBI and DDBZ at DNA Data Bank of Japan. And these three databases actually deposit the primary sequence of data, and they are also synchronized data. The primary sequence database are synchronized, and every sequence receive a unique identifier. That means a unique identifier can be find out the one different type of database. So if you deposit some data, you can find out a number like they're called accession number, and by this accession number you can find out your data. Or if you want to target something, so if you know the accession number of this gene or of this protein, you can find out this data from any of these three databases, and they daily synchronize their data. And now I try to say something about the software and tools. There are many software used for the bioinformatics study. So here we just try to introduce you some basic software and tools. So bioinformatics company software tool for bioinformatics range from simple command that means line tool to more complex that means graphical program and standalone the web service available from the various bioinformatics company or public institution. And some of them are also payable, or you have to pay for do some special research. But here have a huge amount of server that you can be used freely for your research. The computational biology tool best known among biologists to be probably BLAST. So actually BLAST, that means this is the first thing. So we will do for our research. This is an algorithm for determining the similarity or arbitrary sequence against other sequence. That means if you have some sequence, you can try to find out what is the source of the sequence, or you can find the similar sequence. You can similar sequence of your sequence. So this can be happen by BLAST, and BLAST is the one of a number of generally available program for doing sequence alignment. The NCBI actually provide this thing. So BLAST actually say basic local alignment search tool. So by using this tool, you can find out the similarity of this data, or you can if you have some sequence, you can you don't know about the organism or about the name of the protein of the sequence. If you try to know this one, you have to go BLAST, and you can find out the original name of the sequence or the similarity of the sequence with others. And it is an algorithm to comparing biological sequence information such as amino acid sequence to different protein or nucleotide of DNA sequence. And BLAST is used for identify library sequence actually. And this is the BLAST, and it's actually it's from NCBI, and BLAST have different types. So like nucleotide BLAST, protein, or other type of BLAST. So BLAST can be used for several purposes. This included the sequence of interest. So identify the species. If you want to identify any species, so if you have the sequence of this species, you can find out the similar species, or you can try to know about the name of the species. So sometimes it not match, so that you can find out some variation of other species. So with the use of BLAST, you can possibly correctly identify a species and or find the homolog species. This can be useful, for example, when you are working with a DNA sequence from an unknown species and locating the domain. So if you want to know the domain in different part of the protein that can be uh, that can be worked for different purposes. So you want to know the domain, you also you have to do the BLAST and establishing the phylogeny. So if you want to make the evolutionary relationship, so and from some organism, and if you have the sequence, you can do by the you can do BLAST, and you can try to find out the phylogenetic tree using the BLAST web page. And comparison also, you can compare the different type of genomic data by these tools. And depending on DNA and or protein sequence as query, or in the database, you choose a BLAST version. So BLAST have different type of version like BLAST N. So actually query is DNA, and the database also DNA. And BLAST P, protein database, actually most usable for molecular research is protein. And BLAST X, that means translated DNA, and database is protein. And BLAST N, protein and translated DNA. And BLAST X means translated DNA versus translated DNA. And another basic things is FASTA. FASTA actually it started in 1985, and this software package is a protein sequence alignment software package. That means it can be alignment DNA or protein, but most of us use FASTA format because this format is most usable for every research. So FASTA format means without any gap, the sequence is organized. So we call this FASTA format, and this FASTA most usable thing. So FASTA package contain program for protein, DNA, protein translated DNA, and order or unordered peptide sequence sources. And not only these things, we can be used in different type of software or different type of website for our research. So here we just show some software's name or some website name that can be usable for our bioinformatics research, like GeneMark, GeneScan, WAS2, and Ambrose. And also, if you want interest about the RNA, so we can use some RNA-based web page. And you can search your sequence for known domain on Interpro scan. So if you want to know any target, if you want to target any domain, you can try to find out by this one. And here I just show some home page. So one for NCBI. So if you try to find out NCBI, so search by NCBI, you can find this page, and you can go, and here have a huge amount of resource or huge amount of data for your research. And homepage for PubMed, if you are try to find out any publication, so this is the, I think, biggest source for publication, that is scientific publication, you can find out from here. And this is the protein database, so protein data bank, actually PDB. If you want to find out any protein data, that means the 3-dimensional structure data on molecules, so you can find from here. And homepage of PDB here. And this is the last. one. And thank you everyone for hearing me. Thank you. শোনা যাচ্ছে আমাকে?
জি স্যার, শোনা যাচ্ছে স্যার। থ্যাংক ইউ।
আচ্ছা, আমার প্রেজেন্টেশনটা এর মাধ্যমে শেষ হলো। এখানে একটা শেষের স্লাইডটা কি শেয়ার করব?
জি স্যার, আরেকবার একটু কাইন্ডলি শেয়ার করেন। সরি, টেকনিক্যাল সমস্যার জন্য আমাদের অ্যাঙ্করের ডিসকানেক্ট হয়ে গিয়েছে। সরি ফর দ্যাট। আর স্যার কাইন্ডলি একটু আপনি লাস্টের অ্যাসাইনমেন্ট স্লাইডটা একটু শেয়ার করেন।
শেয়ার করা আছে।
ওকে, এটা হচ্ছে অ্যাসাইনমেন্ট সবার জন্য। The assignment হচ্ছে what is BLAST? Write the uses of BLAST. How could you find the function of the unknown gene using BLAST? এটা সাবমিশন করতে হবে 24 তারিখ রাত 12টার মধ্যে। আর সাবমিট করতে হবে research@biopc.org ইমেইলে। এখন হচ্ছে কুইজ হবে আপনাদের সবার জন্য। তো আমি ভলান্টিয়ারকে বলবো চ্যাট বক্সে কুইজের লিংক গুলো দিয়ে দেওয়ার জন্য। টাইম লিমিট হচ্ছে 10 মিনিট। আপনারা 10 মিনিটের মধ্যে কুইজ গুলো সাবমিট করতে হবে। আর এই কুইজ গুলো এবং অ্যাসাইনমেন্টের উপর বেসিস করে আপনাদের টপ 10 কে সিলেক্ট করা হবে। এবং ফাইনালি লিংক গুলো দেওয়া হচ্ছে, আপনার লিংকে ক্লিক করে ঢুকতে পারেন। মাল্টিপল লিংক দেওয়া হবে ভলান্টিয়ার, আরো কিছু লিংক আছে, লিংক গুলো একসাথে দিন। আর সবাই সেম লিংকে না ট্রাই করবো ডিফারেন্ট লিংকে ঢোকার জন্য। আর লাস্টলি Amit Kumar Datta sir, thank you very much for this nice session, and it was a wonderful session indeed. আপনি খুব সুন্দর করে সিম্পলি হচ্ছে বেসিক বায়োইনফরমেটিক্স আমাদের সামনে তুলে ধরেছেন। যেহেতু আমাদের এইখানের ম্যাক্সিমাম স্টুডেন্টই হচ্ছে বিগিনার, ভেরি বিগিনার, সো আপনি যেভাবে সাবলীল ভাবে বেসিক্স অফ বায়োইনফরমেটিক্স বোঝানোর ট্রাই করেছেন, দিস ওয়াজ জাস্ট আউটস্ট্যান্ডিং এফোর্ট এভার। And thank you again, sir. Thank you very much.